My name is Tomosaka of Surface Sciences, and I am a senior technical educator. Now I'm going to introduce our two robotized automated protein synthesizer. One is Protemist GT2 and Protemist XC. This is the GT2 here. This slide explains the features of the Protemist GT2. This robot can do everything for transcription, translation, and purification. So, input DNA, output purified protein. And this robot has two modes for translation. One is 1.2 mil reaction or 6 mil reaction. And translation method is a bi-layer method. And this robot can do purification, and the method is batch affinity purification for his or GST-tagged protein. And this robot has an incubator to control the temperature for translation and purification from 4 degrees to 37 degrees. And running time is 35 hours, including the purification process. And this robot is suitable for screening, functional analysis, assay development, and antigen production for antibody. And this protein can produce up to 500 microgram in, per reaction in 6 mil reaction, and 100 microgram in 1.2 mil reaction. This is the bi-layer translation system explained by Zach. This slide explains the integral layout. I'm going to talk about this later. This is the reaction card, the container for translation and purification. Originally, we produced it. As you can see, there are a filter bottom of the container. I'm going to explain what is the filter, what kind of function this has. This slide explains the purification process. Translation is done in the reaction cup to this container. After the translation reaction, for the purification process, add resin into the reaction. And this robot has a function to shake the container to continue the binding reaction. After the binding reaction, the incubator is tilted, and pipetting tip is going to be inserted into the nozzle of the reaction cup and suction the liquid. The liquid is a flow-through fraction, and the express protein which has a GST tag or his tag is binded to the resin, and the resin is retained in the container of the cup, inside the cup, because they are filled. After the suction of the flow-through fraction, add wash buffer to wash the resin, then remove wash fraction, and last add elution buffer to elute the tagged protein. This slide is an example of the result of protein expression and purification by GT2. In this case, we produce express and purify 6 proteins with his tag, and after the expression and purification by the GT2, protein yield around 200 microgram with a purity of around 80%. This is an example of the GST-tagged one. The protein yield is around 150 microgram with a purity of more than 80%. University of Wisconsin-Madison has a GT2, and they use this robot for NMR analysis by HSQC. This is the detail of the protocol for protein synthesis and purification for histone protein. Transcription is done at 37 degrees for 6 hours, and translation is done at 15 degrees for 50 hours. And the purification process is a batch affinity purification, and the protocol is a standard one. This is a case of a GST tag. This slide is a list of reagents for transcription and translation. We sell all reagents required for transcription and translation except water and creatine kinase. Just you need is to prepare the plasmid DNA. This slide is a list of reagents for purification. We do not sell purification reagents, but you can prepare by yourself because the all reagent is a very regular one, not a specific one. This slide is a list of consumables required for ET2. The first two items is we are sole supplier for the pipette tip and reaction cup, but other items you can purchase in your country. This is very regular. This slide is space requirement for inflation, but as you can see, this unit is not so big. I think this robot can be placed on your bench. Next, I'm going to introduce another robotic protein synthesizer, Protemist XE. This is XE. The future is this. This robot can do only translation process. So you need to do transcription by yourself manually. And the translation system is a filter and feed method. And the running time is 24 hours or 48 hours. You can choose it. And the reaction scale, you can choose from 10 mil to 40 mil. And this robot is small, so that fits into an incubator to control reaction temperature. And this robot is suitable for structure analysis and antigen production for antibodies. Using this robot with high performance with jam extract 7240, in case of the 10 milliliter reaction, you can get 100 milligram protein in case of the like easy to extract protein like a GST. This is an explanation about the filter and feed translation system. And this robot uses WEPRO filter. Through this filter, translation buffer is extended every 15 minutes, and every 8 hours, mRNA is added to continue the translation. This is a detail of the protocol for protein synthesis. It's very complicated, but robot automatically does. So first, mRNA replacement is mRNA is added every 8 hours. After the buffer extends 2 cycles, and for translation cycle, the translation buffer is extended every 50 minutes. And one cycle, it takes 8 hours for one cycle, so that for 48 hours translation, repeat this cycle, 8 cycles. This is an example of the protein synthesis. This is the protein is GFP, green fluorescence protein. As you can see, the protein is yielded gradually increased, but it reached plateau in 48 hours. This is first published article related to Protemist XE. The Dr. Bibb is a group of Dr. Wisconsin Madison. The title is Robotic Large-Scale Application of Cell-Free Translation to Structure Study, including membrane protein. This is the first article that related to Protemist XE. This slide is a list of reagents required for protein synthesis. You need preparation water, plasmid DNA, chromochemical polymerase, creatine kinase, but all other reagents we can produce and we can have for use. This is a list of the consumables. first item is the Turing harness. This is our original one, but all other consumables are available in your country. Specific comment is this robot is very small, as you can see. So